And welcome to SAM Biology class. Friends, today we shall discuss about S1 mapping. S1 mapping, also known as S1 nuclease mapping, is a nuclease protection assay. This technique is useful for detection and quantification of RNA species in a complex mixture of total cellular RNAs. It is an extremely sensitive procedure, also used for mapping external and internal junctions such as transcription initiation and termination site, and also mapping intron-exon boundaries in RNA molecules. This technique can also be used to discriminate between closely related targets by using probes designed to span the regions where the related genes differ the most. Based on the method of Burke and Sharp, S1 mapping has undergone many refinements over the years. This technique utilizes S1 nuclease, which is an endonuclease generally isolated from Aspergillus oryzae. This enzyme is a monomeric glycoprotein having a molecular weight of 38 kilodalton. It requires zinc ions as cofactor for its activity, and it functions optimally at an acidic pH between 4 and 4.5. Principle: S1 mapping requires a relatively detailed knowledge of gene structure and sequence data of the first exon and several hundred bases of the upstream sequence. The enzyme S1 nuclease digests only single-stranded DNA or RNA. So when a duplex of DNA and or RNA having single-stranded overhangs or unhybridized internal loops is treated with S1 nuclease, these unhybridized regions are digested away. The fragment that remained after digestion represent the regions of identity between two strands of the duplex. So when a strand is labeled at one end, then the length of the labeled fragment remaining after hybridization and digestion reflects the point on the probe where the two sequences diverge. This is the principle of S1 mapping of transcriptional initiation site. Now we shall see as how S1 mapping is used to identify the transcription initiation site. The DNA carrying the suspected transcription initiation site is first cloned into a suitable plasmid vector. The DNA is then digested with a restriction enzyme yielding fragments which contain the presumed transcription initiation site. A more recent approach is polymerase chain reaction, which avoids the need for cloning. In either of the cases, the generated DNA fragment is double-stranded, which has to be denatured, that will give single strands. An alternative is to clone the DNA strand into M13, which is a cloning vector that will generate single-stranded DNA directly. The single-stranded DNA is labeled at its 5' end by using phosphorus 32. This 5' label DNA strand contains the suspected transcription initiation site. We have to find where the initiation site is. So first of all, The 5' labeled DNA strand is hybridized with the mRNA. After hybridization, the next step is digestion by using S1 nuclease. As you know, S1 nuclease digests the single-stranded DNA and single-stranded RNA. So after digestion, we are getting a hybrid which consists of a DNA strand and a RNA strand. The DNA strand is radiolabeled at its 5' end. Here we are having two types of labeled DNA strand. This is the labeled DNA strand before digestion. For example, this is the transcription initiation site. The second type of strand is the one that remained after nucleus digestion. So this labeled DNA strand is protected by electrophoresing the labeled DNA strand before digestion and the strand after digestion, along with size standards in separate lengths by polyacrylamide gel electrophoresis will help us to identify the size of the labeled DNA strand before digestion and after digestion. After that, we can determine the difference between the size of the labeled DNA strand before digestion and after digestion. This difference will tell us about the position of the transcription initiation site. Similar principles can also be applied to determine the position of transcription termination site. In this case, the DNA fragment should have the suspected transcription termination site. This technique can also be used to map intron-exon boundaries. So friends, this is about S1 nuclease mapping. For more updates, please subscribe SAM Biology class. Thank you very much.